GI hormones. Hormones throughout the GI system, gastrointestinal system. So here in this lecture, we will be listing the hormones which are important. Many hormones are there, but we will select few and we will discuss that one. So important hormones which are related to the GI motility, digestion and digestion plus digestion in the GI tract. These are the we will discuss in this lecture. And the functional significance of these hormones, the one which we will be discussing. What are their importance in the GI tract? We will describe that one. And what is the integrative role of these GI hormones? How they are helping each other? Okay. So these are the important hormones which are present or discussed in the whole physiology of GI system or gastrointestinal tract. First one is gastrin. Gastrin, from where it is released?
Stomach.
From stomach, as the name suggests. Then secretin, then CCK, that is cholecystokinin, motilin, somatostatin, GIP and VIP. I will show you the full form. So these are the main hormones. Other than this also present, but we will discuss only these. Gastrin, secretin, CCK, motilin, that is CCK is cholecystokinin, motilin, somatostatin, GIP and VIP. So first of all, gastrin. Gastrin is produced by which cell? G cells in the stomach. It is coming from stomach, from the G cells. What it will do? It will stimulate the release of hydrochloric acid as well as pepsinogen in stomach. This is one function. Second function is it is responsible for increasing the motility of the gastric motility. Then it also stimulates the ileal motility, ileum, small intestine. We have seen yesterday that when the food is still in the stomach, the ileum or the small intestine will show the different movements. Okay, because of gastrin which is released from here through blood, it will come and act on the small intestine. So it will cause gastroileal reflex. Okay, gastric and ileal motility. It also causes relaxation of ileocecal sphincter. So once it comes here and the food is to be passed, it causes the relaxation of the ileal sphincter, so that whatever the movement is happening, it will pour the chyme here in the large intestine. Okay. It also induces mass movement. Mass movement in colon. We have seen just now the colon will have movement which are called as mass movement. This is because of gastrin acting on the colon also. We have seen gastrocolic reflex. Gastrocolic reflex also. So gastroileal reflex and gastrocolic reflex, two things will be there. One is autonomic nerves as well as the gastrin. Gastrocolic reflex. Mass movement in the colon help to move the content of the GIT on arrival of a new meal. So it will push like a cleaning the content so that the new meal should occupy and will start the new process of digestion. So this is in short about gastrin. You have to read it from the books about the details. I have summarized here in few lines or few points. From where it is secreted, what are its function in the stomach, what is its function on the intestine, what is its function on the small and large intestine. Second hormone is number 2 is secretin. From where it is coming? From small intestinal cells. Yes, from they are called as S cells. S cells from the small intestine. When the stomach acid is coming in duodenum, little amount of stomach acid which is poured or the content is poured in the small intestine in the duodenum. the S cell will get stimulated and they will secrete secretin. What is its function? It will inhibit the gastric emptying. It will be opposing the gastric emptying and delay the acid content to reach to the duodenum so that the mucosa should not be damaged by the stomach acid. It also inhibit the gastric acid secretion. So whatever secretion is happening in the stomach, so from here the secretin is coming and it will inhibit the gastric acid secretion or gastric secretion. Maybe it is inhibiting also gastrin little amount. It act on the pancreatic duct. It act on the one another function is it is acting on the pancreatic duct to cause what is the role? To give large volume of watery and alkaline secretion to neutralize. Basically, its function is to neutralize the acidic content which is poured into the small intestine by two ways: by decreasing the emptying of gastric acid, gastric content, as well as by inhibiting the gastric secretion, and the third by increasing the secretion of bicarbonate watery secretion from the pancreas. It stimulates the secretion of soda bicarb rich bile also in the liver. So secretin function is secretion of alkaline pancreatic secretion as well as the alkaline bile secretion. Okay. So it is acting on two places, even on the liver also. It is act here, it acts here to give alkaline secretion, NaHCO3. Third hormone, CCK, or the full name is cholecystokinin. Cholecystokinin. Remember kinin. Oh, sorry. Cholecystokinin, kinesthesia or movement. CCK is released from, again, duodenum in response to mainly fatty meal or fatty undigested food from the stomach. If food contain more fat, there will be more CCK will be released, and to a lesser extent, a little amount of proteins. What are its action? Its action are inhibition of gastric motility. Both CCK and secretin both will be inhibiting the gastric motility. They stimulate pancreatic acinar cell. Now here, not ductal cell. They are acting on acinar cell to secrete pancreatic enzyme. Here it will be more towards the digestive enzyme, not as in secretin. Secretin was more about the liquid alkaline solution, soda bicarb from the ductal cell. Here they are secreting the digestive enzyme like amylase, lipase, trypsinogen, chymotrypsinogen. So it is stimulating the secretion of digestive enzyme from the pancreas. Also, as the name suggests, kinin, it is causing contraction of the gallbladder. Gallbladder will contract. So what will happen? There will be bile will come out. Relaxation of the sphincter of OD here when it is opening. So it will be relaxed and the bile will come into the duodenum. This is the function. So it will cause contraction as well as relaxation of the sphincter. Another function other than digestive function, CCK has got a very special function, key role in food intake or satiety. When CCK is there, it will make the person as if he is full or not having hunger, or it will be satisfying that the food is okay, finished. No need to more eating more. So CCK is another special function. It will regulate the food intake. Please make this is another important function of of CCK as a role in satiety, making the person as if he has taken enough food. Fourth hormone, motilene. It is a polypeptide in nature secreted by EC cell, enterochromaffin cell, and M0 cells in the stomach as well as in the small intestine as well as in the colon. So it is coming from three places: stomach, small intestine, and colon, from the enterochromaffin cell. What it causes? It causes the contraction of the smooth muscle movement in the stomach as well as in the intestine. Its level increases during interdigestive state. Between the two meals. The secretion of motilene will be more. Okay. So if you have taken breakfast here, and here lunch, so in between somewhere there will be release of motilene between the meals. It causes the movement or contraction of the smooth muscles in the stomach and intestine. This is the function of motilene. Okay, the fifth one is somatostatin. Just remember, somatostatin is maybe most of the places it is inhibitory. See here, it is secreted by T cell or delta cell in the pancreas. We have seen pancreas cells are alpha, beta, and delta cells. So delta cells or D cells in the pancreas secrete somatostatin. Also, some D cells are found in the GI mucosa, which will also secrete somatostatin. Somatostatin is inhibitory to secretion of gastrin, VIP, GIP, secretin, and motilene. So once somatostatin will be released, it will cause inhibition of gastrin, inhibition of VIP, vasoactive intestinal peptide. It will inhibit GIP, inhibit here also. It will inhibit secretin also, and it will inhibit motilene also. So that's why I told you it is mostly inhibitory hormone. Why it is secreted, or who will stimulate the secretion? The acid in the lumen of the intestine. If there is an acidic content found in the intestine, it will increase more and more somatostatin secretion. It acts probably by paracrine fashion. Paracrine, not by endocrine. Endocrine means, suppose this is a gland. If it is secreting something, it will go to the blood. This is endocrine. But here it is acting through paracrine. That means if these are the cells or glands which are secreting somatostatin, it will act on the nearby tissues. Here also some other tissues. This is called as paracrine. Nearby, parallel to or in vicinity of that area, whatever organs comes or whatever cells come over there, somatostatin will be acting. Okay, what is GIP? The full form of GIP is glucose dependent insulinotropic peptide. G, I, and P. Glucose dependent insulinotropic peptide. Previously, the name was different. Gastric inhibitory peptide. They were calling it as gastric inhibitory peptide. Here it is given. Because this role is minimum. The other role is more. That's why the name is changed. Previously, it was known as gastric inhibitory peptide, but it is currently known as glucose dependent insulinotropic peptide. So it depends upon the level of glucose. It will act on the insulin secreting cells, and it is peptide in nature. This is the meaning of GIP. So GIP is released from, again, duodenum. It stimulates the release of insulin because it is acting on the cells. and it is stimulated by presence of meal, especially glucose. If sugary meal is there, then GIP will stimulate, will be released and it will act on. Suppose you have taken a sweet meal, sweet meal, not sweat, sweet meal. This will cause release of GIP, not from here, sorry, from the duodenum. The GIP will be released, and this GIP will act on the beta cells in the pancreas to stimulate release of insulin. So what insulin will do? This glucose, because of insulin, will be absorbed. Will be absorbed. It inhibits, another function of GIP is inhibit gastric motility. That's why previously they were calling it as gastric inhibitory peptide. Okay. The last one is VIP. Not very important person. Not very important person. This is vasoactive intestinal peptide. Now, as the name suggests, it is acting somewhere on the blood vessels and in the intestine, and it is peptide in nature. So VIP is found in the nerves in the GIT, the nervous tissues, enteric nervous system. You will find the release of VIP. It stimulates what? It stimulates the secretion of electrolytes and water. Other actions are relaxation of intestinal smooth muscles, including sphincter relaxation. It will inhibit gastric acid secretion, and it potentiates the action of acetylcholine in the salivary gland. It will enhance the action of acetylcholine in the salivary gland. Apart from GIT, VIP is also found in brain and blood and autonomic nerves. Apart from the GIT, VIP is found, or vasoactive intestinal peptide is found in brain cells also, neurons, and the blood also, because it may be mixing with blood, and also from the autonomic nerves. Okay. So this is all about the hormones of the gastrointestinal system. This chart or this table is about the whole process of digestion. What is happening by region? Like in the mouth, which type of motility is here? Let me push it down. So in different parts of the gastrointestinal system, which type of motility is happening? Which type of secretion is happening? Oh, sorry. Sorry. Which type of digestion is happening and which type of absorption. So this is a summary of, if you gone through all the slides, just read all the lectures, yani. Just read this chart or table. You will be revising everything. In which area, which type of motility, and which type of digestion, and which absorption, and which secretion is happening. Okay. Similarly, second chart is about the food particle. For carbohydrate, in which part, by which hormone, by which enzyme the digestion is taking place, from which gland it is coming out, and its site of action. Similarly for protein, similarly for fats. So if you revise while after you study everything, if you want to quickly revise the things, you can refer to these two tables. With this, we come to an end of the lecture number 10. Thank you very much.